Good morning, everybody. I'm John Mann, and it's my task and pleasure today to MC Grand Rounds. Anyway, there aren't too many announcements. Actually, there's only one announcement. Next week's speaker. This is a first for a Grand Rounds announcement, as far as I can see. Nathan Fox, distinguished university professor at the University of Maryland, is giving a talk entitled TBD. Sorry, can't make this stuff up. Today we're going to have the Waelsch lecture, since this is an endowed lectureship to honor the memory of Heinrich Waelsch. It's always good to be reminded as to who Heinrich Waelsch is. And this is his picture. He was born in Austria and trained there. He fled to the United States, escaping from the Nazis. He was a man of broad vision and interests. He anticipated the modern integrative approach through the development of collaborative relationships with investigators in other disciplines in terms of studying the brain. He realized that the brain is an enormous challenge. I think it's probably true to say that even today, as much as we've learned about the brain, we are constantly underestimating the challenge of really finally, getting a more complete understanding of what the brain is all about. Even in those days, he authored 300 scientific articles, which is a prodigious accomplishment. He died prematurely at the age of 62, and he was the professor of biochemistry here at Columbia University and the chief of pharmacology at the New York State Psychiatric Institute. He really kind of was one of the pioneers of the field of neurochemistry. He was instrumental in the creation of an organization, IBRO, which was one of the prime movers in terms of scientific organizations in this field, and now to a significant degree superseded by SFN. He founded the Journal of Neurochemistry, probably not as prominent now as it used to be, and was his managing editor for a number of years. His own interest was in the area of glutamate, GABA, etc. And of course, that was relevant for conditions like mental retardation and epilepsy. Here are the previous Waelsch lecturers. I'm going to ask people to write these down at the end of the presentation today. But you can see this is an illustrious group. It's a tremendous honor to be on this group. And that brings me to our 2018 Waelsch lecturer, David Van Essen from the Washington University School of Medicine. David Van Essen trained at Caltech, where he got his Bachelor of Science. And then he got his PhD at Harvard. He basically moved after a while back to Caltech, where I guess you moved from the advanced from an assistant professor up to acting director and full professor in the Division of Biology and acting director of the Computation and Neural Systems Program. I'm not sure what that was, but it sort of suggests a trajectory in your career, which I found very interesting in terms of reading your publications. And I thought your thesis title was interesting. effects of electrogenic pump on signaling in leech sensory neurons. You know, I've always felt that it really doesn't matter what you study when you're doing your PhD, so long as you learn something important about methodology. Because ultimately, the principles of the methodologies that you apply and the vision that you can acquire of the potential of tools, of research tools, are what are going to decide whether or not you're going to be able to answer any significant research questions for the rest of your career. Anyway, this electrophysiological beginning evolved in an interesting way. David Van Essen has headed this human Connectome project, which has been a huge investment by our field to try and understand how the brain works. And in 1980, I saw the first emerging of this with his paper, two-dimensional maps of the cerebral cortex. And then there's a progressive series of papers which indicate an increasing engagement with understanding the structure of the human brain as a strategy for, you know, basically how does it work, and ultimately why does it and how does it go wrong. And then if you look back at the Waelsch lecturers that you see on our board here, you'll see that we've had a number of individuals that have used the same kinds of, that have tried to address the same kinds of questions at a cellular level, a more systems neuroscience level, a circuitry level, et cetera, et cetera. I want to particularly draw your attention that we had Jeff Lichtman here in 2014. And what did Jeff Lichtman show you? He showed you he's mapping the brain, but at an electron microscopic way, so that you actually see every single synapse of individual neurons, how they're connected together. At the other end, you can start with the whole brain and start to look at that in more detail. This is like two bodies of scientists tunneling towards each other through solid rock. Eventually, those two tunnels hopefully will meet, and we'll learn a lot more about what's going on. So David has had many awards, but he gave the most fantastic talk at the ACNP on the findings from the Human Connectome project that he has guided. And I think we're really fortunate to have him to come here today to talk to us about this. And I'd like to invite David up and present him with a plaque. This plaque reads, the 2018 Heinrich Waelsch Memorial Neuroscience Lecturer presented to David C. Van Essen by Columbia University College of Physicians and Surgeons, Department of Psychiatry, Division of Molecular Imaging and Neuropathology, in recognition of outstanding contributions to the field of neuroscience, February 21st, 2018.
Let us switch gears here. Here we go. So thank you, Dr. Mann, for that generous introduction and for the opportunity to speak to you about my scientific passions and to follow in the footsteps of a truly distinguished set of previous Waelsch lecturers. So cortical function and connectivity and structure is a long ways from leeches, but it has a thematic unity because in that thesis work I did actually study the structure and function of individual neurons. So now we're going to go to a systems level. Let me start by assuring you that I have no financial interest to disclose. But I do want to disclose a passionate interest in psychiatry and those disorders associated with dysfunction of the brain, not because I'm an expert, I'm not at all trained in psychiatry, but I can provide you in the coming 50 minutes with a neuroscientist perspective on what neuroimaging may have to offer. So I hope we can all agree that psychiatric disorders are in some deep way perturbations of brain circuits, but we still don't know to what degree they are reflected in changes or perturbations in anatomical connectivity, in functional connectivity that we'll talk more about. There's more indirect but informative. Are these changes in local circuits versus long-distance pathways, or probably some combination of the two? And to what degree are they generalized across the whole brain versus localized to specific brain regions? Unfortunately, I have to assure you that we will not answer these questions, but I'll show you a pathway forward using better tools for macroscopic brain imaging in humans as well as animal models, and looking after elusive aspects of individual variability in shape, function, and connectivity. So let's start by giving a little bit of background about genes and brains and cartography and connectomes and animal models. But our main agenda will be on this human cerebral cortex, and we'll get to that later. So regarding genes, I find it quite remarkable, this basic observation that our genome consists of a little more than 3 billion base pairs, and it's about the same number in mice and other mammals. And if we look at the components, the genes themselves, by best estimates, as I understand it, there are a little more than 20,000 in different mammalian species. So despite that commonality at the genomic level, we of course look at brains that are wildly different in size between mice and humans, and in sheer number of neurons, millions versus billions. And if we look at the neuroanatomical equivalent of the genes, the fundamental parcels of the brain include hundreds of subcortical nuclei and subnuclei, but we don't have an accurate reckoning in either species yet. If we look at the cortical areas that will be our main focus, I'll show you evidence for a little more than 40 cortical areas in mice. And I'll raise the question that has been persisting for over a century: are there about 50 or closer to 200 in humans? And I'll come to the chase with an argument that we're favoring the larger number, but we won't get to that for a little while. When we look at the differences in brain size, besides the 4,000-fold sheer difference in mass between humans and mice, our brains are several times larger than our nearest living ape relative, the chimpanzee. It's about 15 times larger than the intensively studied macaque monkey. It's about 200 times larger than the little marmoset brain, which is emerging as another non-human primate animal model. The other thing that's evident at a glance is that the cortical folding patterns range from very smooth to extraordinarily convoluted, and these convolutions are what made me a cortical cartographer when I started working on the macaque monkey more than 4 decades ago. So as Dr. Mann indicated, my introduction to cortical cartography was to make flat maps of the monkey cerebral cortex, analogous to flat maps of the Earth's surface, but we couldn't use computers to do the job. They were nowhere near up to snuff at the time, so we used literally pencil and tracing paper as our starting kit for making these flat maps, on which you see the buried cortex is shown in gray. But Dan Feldman and I, in 1991, generated a parcellation drawn from information in the literature that provided evidence for nearly 80 cortical areas in the macaque based on differences in function, architecture, connectivity, or topography, a fourfold fact-based approach that gave answers for a few areas that were consistent across all 4 of these modalities or approaches, such as primary visual cortex and area MT, a motion processing area. But for most of the other areas, the borders were fuzzy at best, and the existence of these areas versus alternative parcellations was highly debatable and continues to this day. When Dan and I looked at the connectivity, again drawing information from the literature, we found evidence for more than 300 pathways amongst the dozens of visual areas known at the time. And we hypothesized that there was a hierarchical organization from early to intermediate to high levels of processing based on the anatomical wiring, the layering pattern of inputs and outputs, but with a lot of feedback as well as feed-forward connections. And we were also able to take the 300 pathways known at that time and plop them onto a connectivity matrix, what we could now call a parcellated connectome, but it basically showed the presence or absence of connections from one visual area to another. At that time, we knew that connection strengths from one pathway to another varied enormously, but we didn't have any quantification, so we simply presented this as a binary connectivity matrix. If we fast forward to the present, we have new and better parcellations of the macaque-monkey cortex. Here is a 91-area parcellation published by Henry Kennedy's lab, but other parcellations show well over 150 anatomically defined parcels. But the truth is that we still have no consensus parcellation for the macaque monkey. But if we take this 91-area parcellation, we can use it to indicate progress in quantifying the diversity of connection strengths. Again, from this study from Henry Kennedy's lab, 29 cortical areas were injected with retrograde tracers, and on average, each area had more than 50 inputs, so much more numerous connectivity than we had realized before. All told, from this initial survey, there were over 1,600 pathways, and we would estimate, if when it's all said and done in terms of all parcel-to-parcel connectivity within cerebral cortex, both hemispheres as well as subcortical structures, a good ballpark number to be thinking about for the macaque is at least 10,000 pathways are part of the overall wiring diagram. The other important point was that the range in connection strengths was staggeringly large, over 100,000-fold range from the strongest to the weakest connections, and for each area they follow an interesting log normal distribution pattern of connection strengths. If we jump shift over to the mouse for just a couple of moments, Andreas Burkhalter's lab has pioneered the parcellation with modern techniques of the mouse cortex. Here's a tangentially sectioned, flat-mounted mouse cortex, and you can see that there are 41 distinct cortical areas in this parcellation, and in work done with Henry Kennedy's lab, using, again, retrograde tracers and quantifying connection strengths over an extensive but still partial connectome, we see that the connectivity pattern is even more distributed, and mammalian cerebral cortex, by these two exemplar cases, is far from being a small world network that it had been hypothesized some years ago. It's very richly distributed connectivity. And in addition, when we look at any given cortical area with repeated injections, the pattern, the profile of connection strength shows some consistency, but some interesting variability that presumably accounts for some aspect of different brain functions. So with that as a background coverage, let's jump over to the human cerebral cortex and look first at variability development and brain atlases. So the starting point for modern brain, human and brain anatomy includes a high-resolution structural MRI. So we get sub-millimeter voxels instead of the conventional 1-millimeter voxels to better delineate gray from white matter. And then we use the free surfer algorithm pioneered by Bruce Fischel and colleagues to capture the shape of the cortical folds. The blue outlines represent the peel surface. The greenish outlines represent the gray-white boundary. We can average between those and generate what we call a mid-thickness surface. We generate an accurate three-dimensional reconstruction, a geometric model, which we can then spin around and manipulate its shape. So here we're smoothing it out and yet preserving an indication of shape features. The darker it is on the inflated surface, the deeper it was in the original brain. If we do this for many individuals, here we show 4 individual healthy young adults, you can see that the folding patterns are indeed strikingly different from one individual to the next. but it's hard to appreciate the full differences with these 3D models until we smooth them out, as you saw in the previous movie, and note, for example, that the central sulcus is robustly present near the top of the brain in all individuals, but in most regions, such as the occipital-temporal cortex here, regions that are involved in higher cognitive functions are extraordinarily variable in their folding patterns from one individual to next. Amazingly enough, this is true even for identical twins. So here's one pair of monozygotic twins on the top, another pair on the bottom. There is some heritability of folding patterns, but the striking thing that hits you in the face is how different the folding patterns are from one individual to another. Another fundamental set of observations relate to the cortical areas that we're going to revisit in detail, but let's start by just noting that well-defined areas such as primary visual cortex, shown in red for 3 examples, vary in size across individuals by twofold or more, and the locations of these areas vary relative to the nearby cortical folds as well. So this brings me to 2 interesting sidebar questions. What is it that makes the cortex fold instead of being a smooth sheet in us and many other large brain species? And why, for that matter, is the cortex a sheet instead of being a big blob like the thalamus or the basal ganglia? Well, we know when cortical folding occurs. It starts at the beginning of the third trimester, where the cortex is relatively smooth except for major furrows, and 3 months later at birth or term equivalent, the cortex is nearly as convoluted in a newborn baby as it is in the adult, even though it's only 1/3 of the size. My thinking about what causes the cortex fold were triggered by observations that Tom Coogan and I had made 2 decades ago, where we noted that the pathways, the connections between V1 and V2 in both directions are established right around the time that folding occurs. A gyrus occurs in between them. And that was my favorite scientific light bulb moment of my entire career, because it led to the hypothesis that if mechanical tension along the axons that connect these 2 areas existed, if these regions are tugging, they would bring strongly connected regions together, forming a gyrus in between, and that's a winning outcome. And sulci, the other type of fold, are essentially, by this hypothesis, the outcome of a losing battle against more stronger competitors. So this one hypothesis of tension-based folding can account for both the consistency in some regions that are dominated by large areas and major pathways, as well as variability that I've already illustrated, where we hypothesize that a balkanized distribution of little cortical areas and diverse connectivity patterns can lead to different outcomes. So mechanical tension does occur in tissue culture if you plot neurons on a dish. It remains a very attractive hypothesis, but it's actually controversial and certainly not proven. And honestly, I think it's likely to be one of multiple mechanisms, along with differential proliferation and cortical buckling, a more mechanically configured operation. But it provides an excellent working hypothesis for what underlies the morphogenesis of brain structures. The other aspect of this to touch upon briefly is, why is the cortex a sheet? It's very thin, and early on it's dominated by the apical dendrites of the numerous pyramidal cells that form in each layer. And the simple but attractive idea is that mechanical tension along apical dendrites could be generating force that pulls the superficial and deep layers close together, keeping the cortex thin, and the expansion of the cortex as dendrites and synapses elaborate would lead to tangential expansion rather than changes in thickness. So this general idea that connectivity is related to morphogenesis and cortical folding gives us a context for thinking about certain brain disorders. So a decade plus ago, we looked at Williams syndrome, a very interesting brain disorder, and showed that the folding patterns are different in Williams syndromes versus control. And perhaps those connectivity differences are related to the differences in behavior that we see in Williams syndrome. Another more common example concerns preterm birth, whereas most of you probably know there's regrettably very high risk for autism, ADHD, and language and other disorders. And if we look at that from a morphological perspective, I've already noted that at birth, healthy infants have a highly convoluted cortex. And in a collaboration with Jeff Neal and Terry Ender, we showed that infants that are born very prematurely, early in the 3rd trimester, if they're scanned at term equivalent, they have cortical folding patterns that show strong abnormalities in many individuals, particularly in lateral temporal cortex, suggesting a selective vulnerability of those regions. So if we think of this and say, well, what's going on in the temporal lobe and what are the functions and connections of those areas, we need to make comparisons across individuals and across groups and across studies. And for that, we need accurate brain atlases. So that lets me shift into the atlasing arena and note that the classic brain atlas was the cytoarchitectonic map provided by Brodmann a century ago. And later in the 20th century, investigators mapped the Brodmann areas onto a stereotaxic atlas. It was literally a book version of the atlas. And then as MRI, structural MRI came into play, group average structural MRIs were generated, which were initially very fuzzy. And then as the alignment algorithms progressed, the group average structural MRIs became sharper, but they're still fuzzy in places. And I'll return to the issue that this is not an entirely satisfactory alignment across individuals because the alignment does not respect the folding of the convolutions of the cerebral cortex. But it does let me set the stage for bringing into discussion mode the so-called traditional fMRI neuroimaging paradigm that started 20 years ago and remains to this day the dominant approach to fMRI and other neuroimaging studies. The starting point is to acquire fMRI data, that's good, but typically it's acquired at a relatively coarse resolution, several millimeters, which is greater than the average cortical thickness. And then to take the individual subject MRIs and register them to a volumetric atlas of the type you see up here, and then smooth the fMRI data, blurring it spatially, and then thresholding it and throwing away a lot of the potentially interesting data, and further restricting the representation by showing typically only exemplar slices in a publication, and then further distilling it by showing the stereotaxic coordinates of the centers of activation foci or other regions of interest, or just saying, well, it's in the middle frontal gyrus or Brodmann area 17 or 19 or whatever. So each of these steps erodes precious spatial information, and all of them can be avoided or improved, and that's what we're going to shift our gears to. So the improvement started with this kind of surface-based analysis I've already introduced, but it came into sharper focus and a more comprehensive alternative strategy with the advent of the Human Connectome project, which I'll introduce now. So the HCP, as the shorthand will have it, started in 2010 with an effort, NIH funded, to acquire data on brain structure, function, connectivity in healthy adult twins and their non-twin siblings. We used better scanners, improved pulse sequences that I'll revisit, and we acquired an unprecedented 4 hours of imaging data from each individual relating to structure, function, and connectivity. We scanned and shared the data for 1,100 subjects on a 3 Tesla scanner at WashU. 200 of the same subjects were scanned at 70 at the University of Minnesota. We got magnetoencephalography data from 100 subjects. We have extensive behavioral and demographic data, as well as genotyping data that at long last will be released next week. It took a long road to hoe for that. But we took all these different modalities and analyzed the data using improved pre-processing pipelines, better alignment and visualization, and a variety of advanced analysis that I'll illustrate a few examples of. Important to our mission was to share the data as well as the methods and tools using 2 databases that I'll touch upon. About 10,000 investigators have been tapping into this data, and already, we're still going strong, but hundreds of publications have emerged from using this data. As well as using the data, the methodology, as I mentioned, is very important. And Matt Glasser spearheaded an effort that we published in Nature Neuroscience to illustrate the 7 tenets of the HCP-style neuroimaging paradigm. So the first two are, one, get as much multimodal imaging data as you can, given practical constraints regarding time and so on, and get the data at the highest quality resolution as possible. Minimize distortion and blurring of each subject's data when analyzing it. Respect the geometry of brain structures, and that boils down to using geometric surface models and surface vertices for the cerebral cortex, and combining that with a voxel-based representation, but only of the subcortical gray matter structure for efficiency of representation. We call that the SIFTI gray ordinate framework. Then we want to align the data precisely across individuals and across studies in ways that I'll illustrate. Analyze the results in relation to an accurate brain parcellation. Then freely share the data, including extensively analyzed publication-related data. So if you or any of your students or colleagues are interested in learning the nuts and bolts of this, we strongly encourage you to take a week-long intensive course. Our fourth version of this will be held in the end of June in Oxford, and you can register online at your leisure in that website. So what do we get for our different modalities of imaging? Let me illustrate briefly with a few examples. So this is a horizontal T1 weighted structural image. And when Matt Glasser came to my lab almost a decade ago, he had already noticed that in the gray matter ribbon, some regions are a little bit brighter than average, other regions are a little darker than average. And in the same subject, a T2 weighted image that has the opposite contrast in general showed the regions that had been brighter are now darker. Those that had been darker in T1 weighted are now brighter. So Matt cleverly did the math, divide and conquer by taking the ratio, then colorizing each voxel according to that ratio, and mapping this onto the cortical surface, where it becomes more evident that the red regions correspond very nicely with heavily myelinated regions that have been shown from classic architectonic studies, including somatosensory and motor cortex, auditory, and early visual cortex. So we call these myelin maps, and we'll return to them in a little bit and show how useful they are. But I want to touch briefly on one other modality that I'll show, distill the essence of it in just one slide, because we don't have time to cover everything in great detail. So diffusion imaging is a powerful approach that relies on preferential diffusion of water along the length of axons, particularly in the white matter. And with the HCP data, we got hour-long diffusion imaging scans giving us high spatial resolution as well as high angular resolution. And we use sophisticated fiber orientation modeling methods to estimate up to 3 fiber bundle pathways within each cortical voxel if it was supported by the data. Then we use tractography to connect the preferential fiber orientations and estimate not only white matter major tracts, but also what we call structural connectivity, diffusion imaging-based inference of gray matter to gray matter connections over long distances. Let me just state that this is a powerful technique, but it is confounded by a number of false positives and false negatives that are related to folding-released biases, as well as the fact that some axons branch, and that's hard to track in diffusion imaging. So it's a powerful method, but I encourage those who use it and who read about it to be very cautious and not over-interpret the data. So let's move on back to a couple more important methodological introductory points. One is what we call multiband or simultaneous multi-slice imaging for fMRI as well as diffusion imaging. And instead of getting data one slice at a time, which literally takes many seconds in a typical scan per image volume, one can excite multiple slices simultaneously and then use parallel imaging multi-channel head coils to extract and infer the distribution of signal in each individual slice. And the multiband factor we used for the HCP was 8-fold for functional MRI, 3-fold for diffusion imaging. This greatly increases the amount as well as the quality of the data and makes us want to use our analysis strategies very efficiently. So another equally important methodological consideration is alignment of subject data across subjects. I already mentioned that the volume-based alignment of individuals to an atlas has challenges, and one way around that is to use cortical surface models. The early versions of what we call surface-based registration used cortical folding patterns to align individuals to an atlas. That works pretty well in regions where the folding pattern is reasonably consistent, but it works less well, and demonstrably so, in regions where the folding is variable and the location of areas relative to those folds is also variable. So the good news is that our colleagues at Oxford, Emma Robinson, led the way in implementing a multimodal surface matching algorithm, which basically gives flexibility and allows multiple features, including functional features such as myelin maps that you saw before, resting state, functional connectivity that we'll return to, but we use those for the alignment process. And we use, as an independent measure of evaluation, task fMRI, to which we'll now turn our attention and set the stage. So Deanna Barch at WashU spearheaded the task fMRI design and analysis strategies. We got an hour's worth of task data from each HCP subject using 7 different tasks. 3 I'll quickly illustrate here. One is in the scanner listening to stories versus doing arithmetic in separate time epochs. Another is looking at movies, one of which shows triangles randomly wandering past another. Another shows them interacting, hugging and kissing and dancing, and the judgment is this a social interaction or not? And then the third task is a working memory task. Is the image you see here matched or not matched to the one you saw 2 preceding images ago? So if we take one of these tasks, the story task compared to a baseline activity pattern, and average the results across almost 800 subjects, we've got large numbers to deal with here, we get an excellent signal to noise, but you see that the strong activations are spatially rather blurry. When we use MSM-SOLC, that means using just the folding patterns alone to constrain the alignment. If we take the same data, same subjects, and use aerial features, what we call MSM-ALL, myelin maps and resting state networks, we get much improved alignment as well as stronger and better signal to noise, including, look at these 2 little divots in lateral temporal cortex that were very blurry when we did the folding only registration. That becomes relevant when we look at the next task, where you see in the social movement versus random, these lateral temporal patches are discrete blobs, whereas they were fuzzed out using the folding only alignment. So this is motivation for using aerial feature-based alignment. And if we look at the results of this for a few more exemplar task fMRI studies, we're going to view them on left and right hemisphere lateral and medial views, as well as subcortical structures, as well as cerebellar flat maps to get the whole brain's gray matter all in one panel. And you've already seen the story versus baseline for the right hemisphere. Here you can see that's relatively symmetric, though not as strong on the right as compared to the left hemisphere, not surprising for a language task. You can also see activations, hotspots in the cerebellum, posterior and ventral. For the social versus random task, you see comparable or even larger activation patterns in the right hemisphere, including strong activation in intermediate and higher visual areas, as well as extensive activation in the cerebellum. With one more task to show, you see a complementary pattern for a working memory task that activates large regions of prefrontal and lateral parietal cortex, and cerebellar cortex as well in different locations. So just with this quick glimpse, you can see that each task engages a complex and partially overlapping network. And this is a fascinating but very challenging domain to decipher, but it's a different picture than just saying one task activates one hotspot or 2 of the brain. When we turn our attention to functional connectivity and resting state fMRI, here's a movie of a few moments in the life of one subject in the scanner looking at a fixation spot. And then when we look at the fluctuating brain signals in space and time, we can take any pair of gray matter voxels or vertices and ask, are they correlated strongly, anti-correlated, or somewhere in between? We can do that literally for 90,000 by 90,000 pairs and get a 30 gigabyte dense functional connectivity matrix as a starting point. And we can probe that in various ways. One is to use what we call seed-based functional correlations and show, for example, that this lateral parietal cortex region is strongly correlated with neighboring strips of cortex on both hemispheres, as well as with prefrontal cortex. We can probe another area, such as the central sulcus, and see a totally different pattern. So that approach is informative, but there are other ways to analyze functional connectivity data that are complementary and very useful. One of them is the independent components, or ICA maps, pioneered by Steve Smith and his colleagues at Oxford, that give fuzzier parcellations or network nodes that include non-contiguous patches. So here is a language resting state network from a group average of 210 subjects showing correlated activity amongst disparate regions of lateral cortex mostly. And this group average pattern can be regressed into individual subjects and giving you individual subject language network maps for subject 1, subject 2, and so on down the line. So this is the language network. We're going to be seeing more of it later. Here is the upper limb somatotopic body representation resting state network, again for a group average and for exemplar subjects. And we're going to, again, revisit this in a few moments. So we can take all of the ICA components. For instance, this is a 25-dimensional decomposition, and generate network matrices, as shown in the lower left here, that represent the correlation, either the full correlation below the diagonal or partial correlation above the diagonal. Partial correlation is arguably better, taking care of cleaning up the data, but these give you maps of functional connectivity of different network regions that can be analyzed for individuals as well as group averages. We're going to use that to compare functional connectivity to behavior, but I need to introduce the behavioral and demographic set of data in a quick coverage here to show you we have diverse phenotypes, many measures that relate to cognition, personality, sensory function, et cetera, as well as data including psychiatric status and substance use. Altogether, we have almost 500 subject measures. And when we take any given subject measure and compare it to the individual differences in functional connectivity, we see some intriguing correlations, but in all truth, most of the correlations do not pass multiple comparisons corrections because there are so many different subject measures. So instead, it's more powerful and informative to use multivariate approaches. Again, Steve Smith et al. Have pioneered the introductory use of this for Connectome data. And CCA, canonical correlation analysis, uses all subject measures to generate one set of modes and all functional connectivity for another. So in terms of the subject measures, this study identified one CCA mode that was distributed with many characteristics along a positive to negative lifestyle atlas. So that include high life satisfaction, good vocabulary and intelligence on the positive side, aggression, bad sleep, and other things that we consider more negative on the opposite end of the spectrum. And when we ask how are those distribution of characteristics correlated with functional connectivity, you can see that the ICA-based node edge strengths were increased for a set of regions that included insular cortex and the so-called default mode network, whereas the edge strength decreases were related to partially overlapping but largely separate region of middle insula and lateral parietal cortex. So that's one intriguing set of observations. I'll just touch upon a paper that just came out in ELOF from Janine Biesterbach et al., showing that the story is even more richly complex when looking at the same type of data, but looking at a different analysis strategy. It turns out that it's not just the edge strength, it's actually the spatial configuration of nodes that differs across individuals and maybe a more insightful way to characterize these behavior-brain connectivity relationships. So just quickly touch upon that and leave it for now that relating connectivity to individual behaviors is rewarding, but it's really a complex thing that is still in the early days of analysis. And I think that analysis is going to be helped by having better brain parcellations, and that's what we now turn our attention to. So you saw Brodmann's map before. He reported 47 cytoarchitectonic areas. The Cecil and Otto Vogt school at the same time in the same institution used myeloarchitecture and reported evidence for upwards of 200 distinct cortical areas. A century later, in 2012, we took high-quality surface map parcellations that identified 52 areas and showed that in the intervening regions there was plenty of room for upwards of another 100 or so areas. So we estimated at least 150 areas, but needed more data to fill the gap. One way of filling that gap has come from functional connectivity that you've seen already. A number of studies have used resting state networks to parcellate with what we call a hard parcellation of the entire cerebral cortex, but they give different outcomes, different numbers of parcels from one study to the next. They don't show as strong a symmetry across the two hemispheres as we might expect, and altogether they are informative, they are forward progress, but there's absolutely no consensus from these kinds of unimodal parcellations. So what Matt Glasser and I wanted to do was take the multimodal HCP data and analyze function, architectonics, connectivity, and topography, the same fact-based approach that worked for monkeys, and to use quantitative analysis of spatial gradients on surfaces to objectively identify borders or transitions between areas, and to do this on a parcellation group of 210 subjects, and then to validate it on completely independently acquired validation subgroups. And you'll see the result of that shortly. But another exciting aspect was that we were able to use a classifier algorithm to parcellate individuals as well as group averages. But let's start with the group average and quickly illustrate how we go about our business here. So this is a group average myelin map, and we're going to focus on these triplet of areas here. Two, the frontal eye fields and premotor eye fields that are moderately myelinated and are known to be related to eye movements, and a lightly myelinated new player on the block that we'll call 55B for reasons that will soon become apparent. When Matt took the spatial gradient of the myelin map and said, where does it change rapidly, there were gradient ridges, we call them, that were modest in visible magnitude, but were highly significant in large effect size in the regions outlined by the white outlines. And importantly, when we took a different modality, such as fMRI task activations from the story versus baseline contrast that you've already seen, the activation is on top, and the task gradient map agreed extremely well with the myelin map gradients. A third modality also agreed. If we take functional connectivity and use a seed location in the language region here, we see correlations with the language-related patch in the black outline, whereas a seed in the eye movement-related area on the upper right shows correlations with the eye movement patches, but not with the language-related patch. And the overall functional connectivity gradients are in excellent agreement with the other modalities. So three modalities give the same answer, and we ended up calling this area 55B because it turned out that half century ago, Adolf Hopp had identified a lightly myelinated patch in this region, and we decided to use the same name. So Matt carefully and methodically applied this parcellation gradient-based approach to all regions of both hemispheres, and we ended up with what you see here, 180 areas on the left hemisphere and a matching set of 180 areas in the right hemisphere. And you can see they vary a lot in size and overall shape, but that's in agreement with previous architectonic analyses. And when we colorize this same map to give a first-order indication of their functional specialization, you can see there's a strip of greenish areas related most strongly to sensory motor function, bluish related to vision, reddish related to audition, and then the bright shades are related to the task-positive network, and the darker shades to the task-negative network. So there's lots of diverse functional information, and indeed, when we look at the profile using the 112 dimensional feature vector we have for each map, we see that the signature for 55B is similar but not identical in the two hemispheres, same for the parietals, the Perisylvian language area, and so on down the line. We also see important evidence for functional specialization within cortical areas. So these are resting state networks showing that the hand representation is very well segregated from the face and foot representation in resting state as well as task data, indicating heterogeneity in connectivity within areas that remain well defined by a different modality of architecture and thickness. So Matt and I looked in the literature and found that of the 180 areas, 83 of them were well enough matched to previously reported areas that we were comfortable using the same name. But for the majority of areas, 97, we either gave them completely new names or we took pre-existing published areas and split them into, for instance, anterior and posterior parts of TE2. And if you want to know the gory details, you can go to the supplemental pages, almost 100 pages and 25 figures in the Nature supplementary results. Or if you want to get fast-track access to the primary data themselves, you can go to the online publication and look in each of the many figures. There's a URL at the end, and you can literally just point and click to that URL, and it brings you immediately to the corresponding data set underlying that exact figure in the BALSA database that we've implemented and tied it to the use of what we call scene files in Connectome Workbench. So you see the pretty picture as a preview. You can agree to data use terms in just a moment, and then you can download the data that underlies this figure and reconstruct exactly on your computer at home what's going on. And there are other data sets. For example, if you want to see the cortical areas, all of them individually labeled, a crib sheet as it were, you can download that by another scene file available in the BALSA database. So what does one do with these parcellations? Well, it's wide open for a variety of approaches and applications. One of them is what we call in the general realm of parcellated analyses. So here is a group average, what we call a dense myelin map, a separate value at each vertex, but we can take the same, the average myelin content in each area and get a much more compact, 300-fold less data file size and get nearly as much useful data to analyze the myelin content and to relate that to other characteristics. We can take the task fMRI data and average that, and here you see the group average task fMRI working memory contrast. This is one of 47 distinct task contrasts that can be accessed at your fingertips. For functional connectivity, we can take these dense connectomes and give much more compact and efficient representations with a parcellated connectome map, which I note is not the same as anatomical connectivity, but it's the best evidence we have about how areas are interacting strongly or weakly with one another. I'll mention very fleetingly in passing that those of you who deal with fMRI data are likely aware that most of the data is noise, and some of it is biased noise and confounds, and there are intense debates within the neuroimaging community as to how to deal with that. Matt Glasser has a paper that's in revision that is available on bioRxiv if you're interested, that uses temporal ICA, analogous but distinct from spatial ICA, to remove global noise but preserve global signal and provide a middle ground of what has been a controversial issue of how best to deal with that. But let me jump over to one of the most intriguing set of observations we found in the Connectome project, which has to do with looking at individual areas and looking at differences across individuals that reflect atypical topology as highly schematized here. So area 55B typically is pointing towards the lateral edge of the upper limb representation, but in some individuals we found it was tilted and shifted in ways that you'll see in reality in just a moment. We introduced this by showing you a group average language network similar to the one you've seen before. Here's a group average myelin map with area 55B lightly myelinated outline. And here you can see 55B pointing towards the lateral aspect of the limb representation. Here is the data for an individual typical subject. 55B lies in the language network, lies right in with the group average. It's lightly myelinated and pointed towards the lateral aspect. Here is an individual where 55B network is well defined, but in this region it's pointing in a offset and tilted organization, and it has a different myelin map, and it doesn't align with the group average. And that's not common, but it occurs in multiple individuals. Here's another example of a tilted 55B. And about 10% of the subjects where we examined this one particular area have an area that's either shifted or split into pieces. So they cannot be aligned by topology preserving algorithms such as the MSM-all method that I've illustrated. But we can get a handle on them using a perceptron-based aerial classifier inspired by work of Carl Hacker and Maurizio Cabeta's lab. And so here you see an individual subject language network and myelin map, and the aerial classifier goes through one area at a time and says, is this little vertex likely to be premotor eye field based on its comparing its feature vector with the group average feature vector? Then it does the same for 55B and all 180 areas, saying what's it most likely to be. Then it takes another pass and generates a hard classification and gets an answer for a typical subject that agrees with the group average. But if we take an individual with a shifted 55B, the 55B probabilistic map gives a very different answer, basically because the neighboring areas are expanded and switched in position relative to 55B. So this gives you a quick indication of how this strategy allows for accurate individual subject identification of all 180 areas in almost all individuals, and shows you a map of an individual match set of areas for one individual or another individual. And if we take all of those individual parcellations by the aerial classifier, we can group them into a probabilistic map of each area, V1, M1, 55B, 46. So you see they're well concentrated, but in some outlier individuals, they're distributed in their positioning. But importantly, we get essentially the same answer when doing this analysis on the original parcellation group or the independent 210 validation group subjects. So it gives us confidence that this classifier is well-trained and works robustly. So in the last few minutes, I want to touch upon just a few diverse topics that tie in thematically but cover some additional ground. So if you're interested in taking your own fMRI data and you happen to have used the kind of free surface-based approach that I've been advocating and want to compare your own data to our multimodal parcellation, this is the kind of question that Michalka et al. And David Summers' lab asked us soon after our paper came out. They had a functionally based parcellation of prefrontal areas, and they said, are these areas the same as our parcellation or different? And Tim Colson in my lab generated the tools and the instruction kit for making that kind of comparison that allowed Sean Tobin et al. In the Summers lab to look closely at their functional parcellation and our multimodal parcellation and say basically they agree reasonably well but not perfectly, but it's a way to integrate across multiple modalities and data sets, and if you're interested, there's a way to tap into that resource for your own use. Let me return to this issue of psychiatric disorders, which I assured you I wasn't going to answer, but let's at least touch upon it. What are we learning from the literature about perturbations of brain circuitry? And there are a number of published studies that I think many in your room know better than I, but one example that I'm aware of that I think is an interesting line of analysis looks at schizophrenia and looks at disconnectivity of specific brain regions, not hyper or hypoconnectivity globally, but more related to specific thalamocortical circuits related to sensory and prefrontal circuits, and Alan Antichevich at Yale has had some success with that and is continuing this analysis using the type of HCP-style data acquisition and analysis that I've mentioned. The core requirements for getting into this HCP-style approach is to get high-quality T1 and T2 weighted scans, field maps for aligning functional data, lots of high-quality functional data, and this is being done as we speak in a number of large-scale projects, the lifespan HCP projects in aging and development that I'm directly involved in. There's a baby connectome project going on in parallel. Importantly, for the current audience, there are 14 projects separately funded in the connectomes related to human disease endeavor. 5 of them focus specifically on psychiatric disorders, and those are going to be part of this freely shared data set as the data come out in the coming years. There's another large-scale ABCD project looking at teenage brain development and maturation. And I want to just shift gears for a moment to remind ourselves of the importance of animal models and interspecies comparisons to frame and put into better context what we're learning about human structure, function, and connectivity. So here's a mapping between monkeys and humans in a movie based inflation, and the result of that analysis provided evidence that in human evolution there's been a dramatic expansion specifically restricted to lateral prefrontal, prefrontal, parietal, and temporal lobe areas relative to the macaque. And it turns out that there are several striking correlations. So if we look at the threefold expansion between adults and infants that I alluded to before, that turns out not to be uniform. The hotspots of developmental expansion correlate reasonably nicely with the hotspots of evolutionary expansion. And they also correlate with cool spots of myelin content that you've seen before. These lightly myelinated cognitive-related regions are much more dominant in human cortex compared to chimpanzee, compared to macaque. And the final thread is at a cellular level. When one looks at data from Guy Elston and other labs at the cellular patterning of dendritic arbors, it turns out that the early sensory areas, the densely myelinated areas, have dense cell bodies and small dendritic arbors that, in contrast to the cognitive, temporal, and prefrontal areas that have much larger cell bodies and dendritic arbors, and indicates that higher cognitive regions are specialized at a cellular as well as a macroscopic systems level way in ways that will continue to be interesting to explore. So to wrap it up, human cerebral cortex has at least 180 areas by our latest reckoning per hemisphere. We can look at that in individual subjects, and we can start comparing species in more quantitative ways in the future. Cortical connectivity is a distributed hierarchy that has very dense networks and a wide range of connection strengths. Individual differences in morphology, function, and connectivity do correlate with behavior, but in ways that are complex and challenging to decipher. We need continued methodological advances to accelerate progress and get deeper insights. And altogether, elucidating human brain circuitry in health and disease is a grand challenge for the century, if not the millennium. So I can't thank enough, even though it will be fleeting, the many colleagues and contributors to the Human Connectome Project and its successor Lifespan Project. Matt Glaster, I've already mentioned, Deanna Barch, Camille Lugerbill, who is co-PI of the project, Steve Smith and Dan Marcus on the informatics project. Other members of my lab have made major contributions. The funding has been extremely important, obviously. And my last set of analogies are taking us up to the 50,000-foot level to remind us that brain maps and earth maps started out in quaint and classical modes. They turned into books for the earth and the brain. They were revolutionized by new technologies, satellite imagery, and MRI-based methods. And they've been further transformed by such extraordinary advances such as Google Earth and Google Maps. And we hope that connectomics will continue to push the frontier of brain mapping and brain exploration in health and disease. So thank you for your attention.
very much. No. Try this. Hello? Yeah. Great. Okay, we have some time for questions. Go ahead.
Thank you. So I noticed that you did a lot of the pioneering work on the 2D representation of cortex, and a lot of people use FreeSurfer to do the same thing today. It would seem to me that your tension-based morphogenesis would almost disagree with that approach because you have these three-dimensional connections that are not following the cortical sheet. I just wonder if you could comment on maybe being concerned about the distance of connections and what might be the appropriate use of either of those approaches.
So I think it's a very interesting question, and one of the corollaries of the tension-based model hypothesis is that it leads to overall wiring length minimization. The network, the system as a whole, is compactly wired just as computer chips are used wiring length minimization to maximize efficiency. So I believe that's very important, and that in every person's brain, your connectivity wiring diagram is efficient and compact, even though it's different from one person to another. So as we change the representation and inflate or flatten these maps, we're changing the apparent connectivity. And what I would say is, with the computerization of this process, we retain information about the actual physical length of distances and even the trajectories of fiber bundles using tractography. So those who want to explore for example, the hypothesis is wiring length indeed minimized in, as best we can estimate, real human cortical and subcortical connectivity. Those are tractable and accessible data sets if you go to the right representation and analyze it carefully. So it's kind of a best of all possible worlds. We use different visualization methods to illustrate different points, but if you're interested in connectivity in the way that you express, one can tap into that information in a reasonably precise and accurate way using the right data and the right models.
Yes.
I have a very specific question and a very general question. The specific question is a number of places in your talk you referred to connection strength. How do you measure that?
Inadequately. It's a great question, and for functional connectivity, it's generally measured as a strength of correlation, but lying under the hood is a lot of twists and turns and complexity. So when I mentioned very fleetingly, full correlation is just basically the correlation between minimally cleaned up fMRI data sets. And if you clean it up more using methods such as partial correlation, you reduce the correlations, but you're still getting a measure that is quantitative. It is a correlation value, but it is far, far from being an anatomical connection strength. And the short answer is it's the best we can do, but one should be mindful that functional connectivity is not an anatomical connection strength, even though I did use that term. And diffusion imaging and tractography tries to estimate the probability of connections and give another highly indirect measure of connection strength, but it too is not terribly satisfactory when you look closer under the hood. So these are foibles and limitations of current methodology, and it's hard to look around the bend for human studies and say, well, how are we going to get more quantitative in living individuals? I'm sure we'll get better by refinements in the methods, but we're not going to get square on hitting the nail on the head. So we need animal models and better anatomical methods to actually measure true connection strength. Does that acknowledge the challenge?
I want to get a general idea of what you mean when you say strength, what you're referring to. I hesitate to ask the general question, but here goes. The whole idea of parcellation is based on an assumption that there are distinct brain areas with sharp boundaries, which seems to me may not be true. There could be functional areas that sort of blend into each other.
It is absolutely a fair question, and Matt and I have debated that more than once. And there are speculations in the literature that this whole notion of cortical areas may be ill-posed if the reality shows fuzzy gradients. I think we are data-driven, and we were persuaded that the transitions that Matt identified past statistical significance easily and past effect size, which we think is also very important to say they are robust. And so we are confident that they will, in general, stand the test of time and be cortical area boundaries when re-examined with better data 10, 20, whenever years ahead. That is not something we can prove until we wait to see if it stands the test of time. And we're confident that some of our areas in noisy regions of the data will undergo some refinements. We're calling this version 1.0, not saying it's the end of the parcellation road. But the question of, are there gradients in organization? The answer is yes. We indeed published in our paper evidence within cortical areas there are essentially, even for cognitive areas, there are topographic gradients of connectivity. So we're not stipulating that each area is a unified whole. Even I showed you the somatomotor areas are functionally diverse and heterogeneous in their connections. So just treating an area as a uniform entity is going to be useful for some analyses, but respecting the internal heterogeneity is going to be very important, and acknowledging that this is essentially a hypothesis, not a proven reality. To go back to your core challenge, I think we should acknowledge that as well. It's going to be, it'll boil down to which approach, which model of cortical organization has the greatest explanatory power, and that'll be a test that will have many dimensions of analysis.
To take off from your Google map, I think the great thing about Google Maps is that you can go down and then you can look around at ground level, and so you can see if that building is a real building and has a door in it and what color it is and so on and so forth. So there is a noble field of endeavor using histology, looking at visual fields and other such structures in the brain that have examined this boundary question, and I wondered whether you thought that a better way of supporting your point.
I certainly think we need more and better data to continue to pound on these questions and to get at the, why do we care about these questions? Because we want to get better understandings of differences across individuals. I also have a vision, a fantasy that a Google Maps equivalent of the human brain would allow tapping in, navigating in an online resource down into any individual's brain and looking at their myelin maps, their cortical connectivity, and exploring whatever you're interested in looking at in a freely navigable, rich way. NIH isn't ready to support us in that endeavor yet, but it's at least as a thought experiment, there is an analogy that's worth pursuing.
Yes.
This is probably a dumb question, but I'll ask it. In terms of the coloration, let's say something is yellow or red or blue, can you give me some idea what the differences are that you get? Because the colors look very sharp. And I'm just wondering what the differences are, if you could explain in some simplistic term for a naive person.
So I think it is a good question, and I'll try to make my answer palatable in the sense that choosing a color and brightness palette is not a trivial matter. And we actually showed many of our results on a particular palette that uses color contrast and brightness contrast hand in hand to share, especially for the colorblind, useful information if you can't appreciate all the hue differences. But we also generally take the trouble to show a mapping between the colors that are vivid or not and the actual values that underlie those by showing a color scale for the chosen palette. And that's generally important, although it does circle back to the earlier challenge. When I say functional connectivity and strength of connectivity, those are squishy numbers. That's just the reality of the current methodology and data. And so being aware of and appropriately critical of where quantification is useful and where it's overstepping what our level of understanding is part of the collective challenge we as a field need to acknowledge.
So I just had another wild thought. Is it possible that you have pushed the spatial mapping almost to an area where it's complete, and that more and more powerful magnets are not going to make a huge amount of difference in the functional areas that you're defining so beautifully, and that to understand the brain better, we have to go. The brain is a time organ that we have to figure that it has to be some sort of a temporal calculus.
So the answer to the first part of your question is I hope very much, and I believe the answer will be no. The bet is on, basically, will higher field strength and further improvements in pulse sequences get us even better data. The 7 Tesla is the workhorse at many institutions. And I would say there's already a lot of evidence that 7 Tesla can outperform everything that I've shown you at 3 Tesla by a significant measure. We are just about to release for the HCP data this 7 Tesla data set that required some extra pre-processing and reprocessing, but our first looks at it show that it is indeed looks to be much higher quality, and some of the questions like the parcellation that we examined can be revisited and see, do we indeed get better answers? So these are testable hypotheses, but there are already published studies showing that 7 Tesla appropriately used can give information about cortical layers, upper versus lower layers, that show interesting functionally relevant differences, as well as information about cortical columns, and other finer-grained aspects of cortical organization. So these are indeed important issues. The time issue that you touched upon at the last of your question is also extremely important and fascinating. Methods such as magnetoencephalography that I mentioned but didn't dwell on, or electroencephalography, do give wonderfully better time resolution, but of course at the expense of dramatic reduction in spatial resolution. Can they be used in a hand-and-glove complementary way? They have been in some studies, and I'm hopeful that they will be better used in the future, but those are also extremely challenging to do incisively, but there's hope for the future, I would say.
Hi, Dr. Van Essen. I was just going to add one bit of an answer to your last one. Of course, with 7T, you also improve the temporal resolution in your image in proportion to the increase in field strength.
Right.
I might just wrap up with one last question, unless somebody has another one. In the brain areas that deal with more basic motor and sensory function, there's been quite a bit of work looking at the boundaries of different parts of those regions in terms of Brodmann area. If you occlude vision in one eye, you see a contraction of the relevant Brodmann areas and an expansion of the neighboring ones sort of filling in the space and other things like that. But in terms of the sharpness of the boundary, the big thing in human subjects is, in humans, is the giant prefrontal cortex, where presumably that's the playground of most of our most sophisticated functions. And we see areas devoted to assessing social cognition signals and areas associated to with the work of decision making, and all kinds of things are, located in bits of this large domain. And I just wondered what your thoughts were about the probability that those functions really map on, how precisely they map onto these different parcellation maps that you're producing. Are they fuzzier or less or not compared to?
Yeah, it's an extremely important question to which I will once again not deliver an incisive answer, but I'll have some brief comments. So, there are countless studies in the literature who said, we found this or that in dorsolateral prefrontal cortex. Well, Matt and I have parcellated dorsolateral prefrontal cortex into, I think it's 12 multimodal areas. And when we look at the feature vectors and say how similar versus how different are those areas, it's a mixed bag. There are different network components that reflect intermediate levels of commonality versus distinct or discrete functional signatures, and those are interspersed in a kind of salt and pepper way within this lateral prefrontal cortex region, and the same for other brain regions. So there are going to be some regions that are very strikingly different in their functional signature, and we predict will be very functionally relevant, including in disease conditions. And any of those, 55B or many other areas that we can point to, having the ability to map them accurately in individuals is, we believe, going to be important to accelerated progress. But one could be a skeptic and say it's all a big blur and it's sufficient to just continue thinking of DLPFC as a big monolithic enterprise. I'm going to bet against it, but the acid test will be, do we find neurobiologically and hopefully clinically relevant, finer-grained, informative analyses using these kinds of high-resolution spatial approaches that I've pounded the drums beat on.
Okay, well, I'd like to say that I want to bet on the same thing. And I want to thank you for a really masterly and wonderful talk. I learned a huge amount today. And thank the audience for their questions. And the residents are invited to have lunch with Dr. Essen and ask him any sort of questions they like. And thank you for coming.